Morning, thanks. And so, by way of context, obviously what we're all trying to achieve ultimately is to advance scientific knowledge. And what we're really talking about here are the infrastructural elements that can support that. And my question is the extent to which the current status quo is optimized towards that goal. And I think a key thing to distinguish between is the extent to which the current system rewards the careers of scientists rather than science itself, if you like. In other words, does what it take to become a successful scientist map onto what it takes to discover new findings that are genuine and robust and reproducible? And so there's been a lot of interest recently in this question of research reproducibility. You may have seen some of these publications relating to cancer biology, psychology, preclinical drug targets. The pharmaceutical industry in particular basically doesn't trust academic research anymore because they can't reproduce their findings, and so much of the published literature to them is essentially junk because it doesn't provide them with any signal against the enormous background of noise that it contains. Psychologists have spent a lot of time looking at this question, principally, I think, not because psychology is any different to any of the other biomedical sciences, but because this is a problem of human behavior. The incentive structures that shape the behavior of scientists away from good science and towards career advancement. So here we have the results of the reproducibility project that published in Science recently, showing the results of an attempt to replicate 100 key findings in psychology that were selected from some top-ranked journals in 1 year. On the left-hand side, on the left panel, you see the p-values for the original studies, and then on the right, the p-values for the replication studies. And on the right-hand side, the same for effect sizes rather than p-values. And the headline figure is that about 1/3 of those 100 findings could be reliably replicated. The definition of replication is obviously non-trivial, but using a variety of definitions, that was the headline message. The vast majority of psychology findings, at least, can't be reproduced. This maps perfectly onto the experience of the pharmaceutical industry when it comes to preclinical science and drug targets. This has been characterized as a crisis, but perhaps it's an opportunity for us to think again about the way in which incentive structures in science are set up and whether they're optimized to actually advance the career of scientists, as opposed to advance science. Here we have a cartoon borrowed from Dante's Inferno by the blogger Neuroskeptic that was published in Perspectives on Psychological Science, and it describes some of this grey area between limbo at the top, where we perhaps just sit on our unpublished data because they're uninteresting to us or we perceive them to be uninteresting to others, through to overselling our results and post hoc storytelling, also known as harking, hypothesizing after the results are known. As an editor, I had a manuscript that was sent back to the authors with the offer of major revisions, and one of the reviewers had said, "We were surprised you hypothesized X, we thought you would hypothesize Y." And the authors came back in their response and said, "Thank you for that comment. In response, we've changed our hypotheses." And the fact was that the system has become so bent out of shape that they thought that it was okay to do that, to retrofit their hypotheses, to go back in time and pretend in the published article that what they had hypothesized was X when it was in fact Y, or vice versa. So it's not so much that they did that, but they thought that it was so okay to do that that they put that in their response letter. P-value fishing, running multiple analyses to see whether we can find that magical p-value less than 0.05, creatively dealing with outliers. There are statistics textbooks that say you should run your correlation, look for outliers, run the correlation with and without those outliers, and then select the significant result because that's more likely to be correct. Completely back to front, but that's in statistics textbooks. And then down into the sort of murky depths of hell where we're doing things that we know are wrong, plagiarism, and then down in the icy jaws of Satan, falsification, of which there are cases, but those are relatively rare. What is more of a concern to me is this spectrum of behaviours that are so commonplace that they have become acceptable, even though they essentially are bad science. So there's a nice example of how this can be achieved in this publication by Simmons and colleagues, published a couple of years ago. They collected real data, and what they showed in an experimental design was that if you play the When I'm 64 by the Beatles to people, they become younger. Not they feel younger, but they are younger. It's an experimental design, so it must be causal, and these are real data. What they show is an abstract that could be presented in two forms, depending how honest you are. The redacted form, which is in bold, and then the unredacted form, which shows all of the different measures that were collected and analyses that were performed and options that were selected and then deselected to arrive at that magical p-value of 0.04 at the bottom. I'll give you a moment to read that. So that analytical flexibility allows you to generate a publishable, in other words, statistically significant p-value from almost any data set, and that's been shown in a number of simulations, which then means, of course, that your p-value is essentially meaningless. This has been shown in the functional MRI literature, in human laboratory studies. This was a nice systematic review by Joshua Karp showing two things. First, on these various measures of study quality and reporting of those metrics on the right, that many of these important features that allow you to assess the quality of a study simply aren't reported in most of those papers, things like the statistical power of the study, particularly problematic for fMRI studies, which tend to be small. But also, because the analytical space in fMRI is so large, because there are so many analytical decisions that you need to make as you work your way through that pipeline, there were almost as many unique analysis pipelines as there were studies. In other words, almost no two studies analyze their data in the same way, which may be that everyone has their own kind of crafted way of analysing data, and to an extent that will be true, but it may also be that people are just analysing their data in a multitude of ways to get a finding. And of course, then it's not a finding, because it's almost certainly not true. This has been described by Andrew Gelman as the garden of forking paths, that we start out with our data and we explore our data. And of course, we encourage people to explore our data. But we also tell them that when you find a p-value less than 0.05, well, that's publishable. And we don't take the denominator into account, the number of statistical tests that we've conducted. It's like searching for a pot of gold at the end of the garden. Once we found it, our p-value less than 0.05, then we begin writing up our results and we get very excited and we start drafting our cover letter to Nature and taking everyone out for drinks. But it doesn't mean, the p-value does not mean what you think it means if you've arrived at it via that pathway. It only means what you think it means if you have a pre-registered protocol, a pre-specified primary outcome, you stick to that, and then you get your p-value less than 0.05. That has been completely forgotten, or was never taught to the majority of people in the biomedical sciences, in my opinion. And that's not, these are not issues of malpractice or bad behaviour. It's unconscious, it's poor training, it's the incentive structures that we work within where there's this prioritisation of statistically significant findings. And another thing that should give us pause is the fact that we're so damn good at science. Because if you read a paper, the data always support the hypothesis, or nearly always, certainly in psychology. 95% of the papers in psychology show exactly what the authors said they were going to look for. Astonishing. So Daryl Bem doesn't need to be doing, the famous psychologist, doesn't need to be doing studies of precognition and the ability of people to predict the future. He just needs to recruit some psychologists. Because we're really good at science. Or we're doing really trivial science, where everything, are men taller than women and we get a small p-value. That name is it's a satire. It's a really good article, actually, and the author is an anagram of his real name, so I'll let you work that out for yourselves. But clearly something's up. And actually we know that because if you look at randomized trials, clinical trials, which are at the very end of a long developmental pipeline from preclinical work through all the different phases of clinical trials, about 50% show efficacy for the treatment. So at the far end of the developmental pipeline, about half of our hypotheses are actually correct. So in that earlier stage of the pipeline, when we're doing exploratory work, should we expect that 95% of our hypotheses are correct? That doesn't seem plausible to me. And the example of the authors that retrofitted their hypotheses to fit the data on the advice of the reviewer, so they thought, supports that view that something's up with the way that we do science. And my view is that this is about incentive structure. So, for example, in the United States, people's salaries are guaranteed by their grant income in many cases. You can have a tenured position in the biomedical research department at an Ivy League university, and 0% of your salary is guaranteed. You have to get grants to pay your mortgage. Now, what kind of behaviour does that incentivize? To what extent does that force people to be publishing papers so that they can secure their next grant and perhaps overly enthusiastically analysing their data to get a finding, overselling their results, aligning their findings with the current political will, if you like, so that you end up in the realms of policy-based evidence rather than evidence-based policy, which we're seeing in certain areas at the moment. And so you see empirical evidence. We showed this for genetic association studies a couple of years before Finelli and Ioannidis, and then they showed this on a sort of larger scale, more authoritative study published in PNAS. We also looked at the average statistical power of neuroscience studies. We've recently updated this to show that this pattern is the same whether you look in psychiatry, neurology, somatic diseases. The average statistical power is about 20%. Studies are too small to give meaningful results, which means, particularly if we're doing exploratory research where the prior odds of the effect being real are low, the likelihood of a statistically significant finding, particularly one with a p-value close to that 0.05 threshold, actually being true is vanishingly small. An average power of about 20%, when we know that and you can see the distribution in the top right-hand corner, we know that conventionally average power should be at least 80%, and now the FDA mandate power of something like 95% for phase 3 clinical trials. So woefully inadequate. Why? Because if I have a £100,000 grant, do I invest all of that in a single publication, which may give me a null result, which will be uninteresting, or do I carve it up into lots of little studies, which will maximise my publication output, and also, because these are low-quality studies, they're small studies, actually maximise the likelihood of me finding something really striking, because low-quality studies are much more likely to generate eye-catching results. Is that what I should do as a rational strategy in order to advance my own career? I would argue yes. But it's not what I should do to get the right answer. And we assume blithely that science is self-correcting, but that's an empirical question. Is it self-correcting? How do we know? Have we tested? Have we checked? And it may be self-correcting in principle when done correctly in an infinite universe, but when done incorrectly over a human lifespan, maybe not so much. We don't know. What we do know is that it's very difficult to convince scientists that they're wrong. How often have you been at a conference arguing about something that you disagree on, where one person has gone, you know what, I've been wrong all these years. Thank you so much for pointing it out to me. It's not human nature. And we become very invested in our own ideas. In fact, we're encouraged to do so. Junior researchers are told, carve out a field for yourself, become known for something, and then do that thing. But of course, that makes it very, very hard for you to say, you know, that thing was wrong. I need to move on, because you're so invested in it. And if you show an ambiguous set of results to two groups of people, an ambiguous meta-analysis, one of those where the results differ depending on whether you're talking about fixed effect or random effect, and show that to methodologists who've got no skin in the game, and primary study authors who've published statistically significant findings in that field, who do you think thinks that meta-analysis supports the position? It's the people who have investment in that particular position. They believe the ambiguous results, whereas the methodologists don't. And the problem is that new researchers coming into this field, because of problems of publication bias, but also citation bias, have no awareness of this. So this is a study by Stephen Greenberg in the BMJ a couple of years ago. The subject matter doesn't matter. Essentially what we have on the bottom left-hand side of that panel are the initial studies that generated sufficient support for that work to move on to animal and cell culture models. And you can see that there were 6 early studies in the early 90s that were critical, 5 that were supportive, but those 5 supportive studies were enough to generate the blue studies on the right, the animal cell culture models. Who do people cite? No one likes bad news. No one likes someone who's a glass half empty kind of person. No one likes the replicators, because that's boring science. So everyone cites the supportive data. No one cites the critical data, apart from that one white line, which is the critical citation saying, well, there was this study, but it was crap. So if you're a new researcher coming in at the animal cell culture model level, you have a completely distorted perception from reading those papers of the literature that your entire work is grounded on. That research becomes invisible. That early research, that critical research, is completely invisible to people coming in at the next level. We've shown this recently for another literature, the serotonin transporter and amygdala activation to threat. 2 main findings here. The first is that abstracts tend to overstate the results. They claim that they've showed evidence for something, but actually when you analyse the data in a sort of harmonised way as you would in a meta-analysis, many of the results are much more tenuous than the abstracts would have you believe. But critically, the studies that fail to show support for this hypothesis don't get cited. So you can see that both within the network of those studies, 21% of them were refutation studies, but they only got 3% of the citations. And in the wider web of science literature at the bottom, 25% of the studies were refutation studies, but only 4% of the citations went to those studies. So again, this myopic view, let's focus on the good news, let's focus on the discovery and the novelty and the advancement, and not do the error checking, not make sure that actually what we're finding out is true. You can see what the impact of pre-registration is here from this recent study showing that prior to the mandation of pre-registration of primary outcomes in clinical trials, all of the studies were positive before that line on the left. And afterwards, the vast majority were null because actually a lot of science is a bit boring. And in fact, one of the studies showed harm, whereas previously none had. And so finally, I've been working with an evolutionary biologist to look at how we can think about the scientific ecosystem. And basically what this figure shows is the proportion of research that is of good value. It's a multiplier of exploratory studies and confirmatory studies, because we need both, further multiplied by the proportion of those studies that are correct, because we don't need lots of novel studies that are wrong. We need some novel studies that are right. And what you have on the axes are the diminishing returns of papers and the weighting given to exploratory findings. I would argue that at the moment, research excellence framework exercises like the REF 2014 that we had in the UK place us at the top right of that first panel, putting a weighting on a small number of papers that are very novel and groundbreaking. And that puts us into the blue, where the majority of that research is not of value to science. What we need to do is move that. We can do that in two ways. We can change those top-down incentive structures by, for example, allowing more publications to count and not placing such a great weight on novelty. That's something that the funders and institutions can mandate. Or, as journals and journal editors, we can change the power of the studies that we require, which is the middle panel, which moves everything up towards the red. And we can, assuming that we're still in the top right-hand position based on the incentive structures, and we can make our alpha level, our statistical evidence requirements more stringent, which is the third panel. So as journals, we can enact change from the bottom up as well to improve the quality of science. And open science is something that I'm increasingly trying to do in my group, not because I think loads of people are going to read it, not because I think the man on the Clapham Omnibus is going to read my article, or that anyone's going to download my data, but the act of making everything open, a bit like open peer review, enforces quality control at every step of the procedure. My postdocs now check their data 4 times, whereas they would previously 3 times, because they know they're going to be made open. And so that quality control is built in at every step of the pipeline. Thank you.